We've now discussed most of Chapter 9. We've covered gene expression, recombinant DNA technology, genetically modified foods, as well as most of genetically modified humans. This last section will highlight the topic of gene therapy. As we talk about gene therapy, there's two main approaches. You can use an ex vivo approach or an in vivo approach. There are several different vehicles for delivering the DNA into the host. We can use viral DNA as well as bacterial plasmids and artificial chromosomes. We'll also discuss how we actually cure different disease states. And lastly, I'll highlight where I think the field of gene therapy is actually headed and using things like RNAi or RNA interference, as well as enzyme replacement therapy. What exactly is gene therapy? Well, it's a means of delivering therapeutic genes into the human body to correct disease conditions created by a faulty gene or genes. So issues are, how do you actually deliver these genes? How can they be targeted to the appropriate tissue? And lastly, are they safe and are they effective? So the key steps in gene therapy are you start with the DNA of interest, the functional DNA. You need to get that into some sort of a vehicle, which is highlighted here in yellow. And this can be bacterial plasmids, they can be artificial chromosomes, they can be viruses. That will then somehow infect the cell, and hopefully the DNA will get integrated into the host chromosomes, and ultimately the gene will be expressed and you'll then make functional protein. So as I mentioned, there's two main approaches to gene therapy. There's ex vivo and there's in vivo. Ex vivo implies that you take it out of something that is alive. So in this situation, cells are actually removed from the patient who has the particular disease. The cell is then treated with the appropriate DNA that's functional, and then you reintroduce those cells back into the patient. The good thing here is that the cells are usually not rejected because they are derived from the patient themselves. The second possibility is in vivo, which implies in something that's alive. Here what you're doing is you're introducing the therapeutic genes directly into the tissues and organs without removing the cells. The difficulty here is that it's hard to deliver the genes to the specifically targeted tissue. So what do these two approaches look like? So here we have the patient. So one option is to do the ex vivo approach, in which you would remove the cells from the patient. You'd grow them in tissue culture. You would then introduce the wild type form of the gene and culture that for a while, and then you would introduce those cells back into the patient. This is in contrast to the in vivo approach, in which you would develop a vehicle that contains the wild type gene, the functional gene of interest, and you'd use that vehicle, in this case a virus, to directly infect the cells, and for this patient specifically into the liver. Some of the different ways in which in vivo gene therapy is done is by injecting the vehicle with the appropriate DNA directly into the tissue. You can also introduce it into the blood or the lymph system. Lastly, you can use something like a biologist gene gun, which we discussed when we talked about genetically modified foods. These can be used for human patients as well, in which you directly introduce the gene of interest, which has been inserted into plasmid DNA. One key vehicle for getting DNA into the cells is using viruses. Viruses are readily able to infect DNA, and so we've alter the viruses, you disable some of the genes that cause various illnesses and diseases, and rather you introduce your DNA of interest. The virus is able to infect the cell. Ultimately, the DNA is hopefully integrated into the host chromosome, and the appropriate gene and ultimately the appropriate protein gets made. So the first example that we know of someone who was treated by gene therapy was a young girl named Ashanti De Silva when she was only 4 years old. She was suffering from something called SCID disease, or severe combined immunodeficiency. She actually had 2 siblings who were also suffering from this condition. For patients suffering from this immunodeficiency, we know that their immune system is highly compromised, and this is due to the fact that they have a defect in just a single gene, the ADA gene, adenosine deaminase. What ADA normally does is it's able to metabolize DATP. So if it is a non-functional gene, what we do is we accumulate massive amounts of the DATP. We know that high concentrations of DATP are toxic to T cells. So T cells are involved in your immunological response, able to help you combat different types of infections. So if the ATP is causing toxicity to your T cells and you don't have functional T cells, then you cannot handle different infections very well. So you're not able to fight off viruses and bacterial infections, and these patients traditionally do not live past their teens. Once they start to spike high temperatures in response to various types of infection, it oftentimes leads to severe brain damage. So how did they treat Ashanti? Well, they used a retroviral vector containing the SCID gene. They used an ex vivo approach, so they removed her cells, and specifically they removed her T cells, and these are easily isolated from the blood. They were then able to use the retrovirus to introduce the functional SCID gene, and these cells were cultured in the lab. The retrovirus caused the DNA to integrate into the genome of Ashanti's T cells, and then these were returned back to Ashanti. So this therapy saved Ashanti's life, and over 24 other children with this immunodeficiency have been successfully treated with gene therapy. The one problem with this type of therapy is that since you are repairing the T cells have a relatively short life, and so you're constantly having to replace those T cells. So every few months, Ashanti would have to go in and receive an additional treatment for the gene therapy. So today she is receiving enzyme replacement therapy, in which she's actually receiving the protein, the functional self. There are some risks to this type of therapy. In 2002, in France, they were conducting a clinical trial on 11 children. 4 of these children ended up developing leukemia, and 1 of those 4 ended up passing away. What we know is that this retrovirus was somehow able to integrate into a different gene that was a transcription factor for the formation of your white blood cells. This led to uncontrolled division. More recently, patients suffering from SCID have been treated with stem cell therapy. So in 1995, the first case of a SCID patient treated with umbilical cord blood was recorded. The cells were treated with vectors that contained the functional ADA gene. So patients then received their own umbilical cord blood cells back. what do you think would be the benefit of stem cells over the current gene therapy? One major benefit is that the body now has a permanent population of cells able to produce the wild type form of this gene. And what we know is after a single treatment, the immune cells in the children were able to produce the wild type ADH enzyme, and they've continued to do so for several years. This is in contrast to Ashanti De Silva, who had to go in several times a year for her treatments using the traditional gene therapy methods. With regards to gene therapy, it's an appropriate method if what you want to do is to introduce a functional form of the gene. But what if what you want to do is actually to inhibit gene expression as opposed to increasing its expression? Well, RNAi or RNA interference is a possible option for those who are overexpressing a gene and you want to inhibit that expression. RNAi as well as antisense RNA can be used to inhibit gene expression. The benefit here is that you can target the inhibition of just one gene, and ideally what we tend to see is you get less off-target effects. You aren't inhibiting lots of other pathways. Our bodies have developed a mechanism by which we can rid ourselves of double-stranded RNA. That's because double-stranded RNA is most often derived from viral particles, so it's a way that our cells have developed to protect themselves. So knowing this about our ability to destroy double-stranded RNA, two recent techniques have been developed, one called antisense RNA and the second called RNAi or RNA interference. So let's say you have a gene that is being overexpressed, which is causing your disease state. This would be true for something like Huntington's disease. So if you have the mRNA that's being expressed, in antisense RNA, you can design a single strand of RNA that is complementary to the gene that's being overexpressed, to the mRNA that is made. And once the complementary antisense RNA binds to the overexpressed gene, no translation will take place and that complex will get destroyed. The double-stranded RNA will get destroyed. With RNAi, it's a little bit more complicated. So what we can do is design a short region that is homologous to the DNA that is being overexpressed. That, and upon entry into the cell, will get destroyed and by the dicer complex. So then at this point we have little short regions of double-stranded DNA. This will then bind to something called the RISC complex. And at this point we just have a short region of single-stranded RNA. If we've designed our synthetic siRNAs to be complementary to the mRNA that's being overexpressed, we now have this small region of homology. And just as before with the antisense RNA, anytime we see this double-stranded RNA, it will be destroyed. So then, theoretically, in either scenario, if you have that overexpressed gene and we design the RNA that's supposed to interfere with it appropriately, we can inhibit the overexpressed gene. RNAi is being used to treat an eye disease called macular degeneration. This affects over 1.5 million adults. What it's due to is excessive growth of blood vessels in the eye, and this is caused by something called VEGF, or vascular endothelial growth factor. So siRNAs have been designed to inhibit VEGF expression, and some 25% of patients have had improvement, and the other 75% at a minimum have had stabilization of disease progression. There are several limitations to gene therapy, the first of which is that These types of therapies are not permanent, but rather short-lived. You have to keep reintroducing the therapeutic DNA into the appropriate cells, and depending upon the lifespan of the cell, you might have to do numerous treatments for a particular patient. These types of therapies can mount immunological responses. If you activate the immune system, then obviously this type of therapy will not be effective. There's also many problems with various types of viral vectors. They can be toxic. They can stimulate that immune response, as I mentioned, as well as inflammation. It's hard to know exactly how much of the gene is going to be expressed and if it's going to be expressed in the appropriate tissue. And gene therapy really isn't doable if it's a multigenic disorder. This is only going to be effective for a gene that is being overexpressed. So one last option besides the gene therapies that I've talked about is enzyme replacement therapy. Here, rather than treating the gene, what you're going to be doing is treating the patient with the wild type enzyme or the protein. These are given to patients by IV. Again, the patient is going to have to be treated repeatedly because over time that protein will be degraded. This is the type of therapy that Ashanti has been using most recently. This also works for a whole host of lysosomal disorders.